IL19 Ghosh, who is an assistant professor in Department of Zoology, Rajiv Gandhi University, Arunachal Pradesh. His area of interest includes biomarker discovery, lower respiratory tract disease, drug resistance in cancer, extracellular vesicles. He has done PhD from University of Burdwan, West Bengal, and he has been a postdoctoral fellow in Kansas City VA Medical Center. He has 2 approved patents and more than 20 publications in reputed journals. Over to Dr. Ghosh.
A very good morning, and at the very beginning I would like to thank the organizers, especially Anju madam and Alok Bharti sir, for giving this opportunity to present my work here. So today I am going to present a part of my work that I did as my postdoctoral fellowship as well as what I am doing in my current lab. And today I am going to talk a little bit about the regulation of triple-negative breast cancer by ETCG, and the entire story will be told with a little twist. Now, firstly, I don't have any conflict of interest in this matter, and so from the beginning of the session, every speaker has told you about breast cancer, and it is very much known that when you talk about breast cancer, the triple negative is the most dire form of breast cancer that we come across. And so I have been working in this field for some times now, and the main issue that came up is basically we want to make breast cancer, triple negative breast cancer, responsive to certain type of hormone therapies. And while doing so, my work is basically focused with a particular group of protein, that is CCM family of protein. Sorry for the bar, it's covering the heading. And this CCM family of protein is a highly conserved secretory protein. And the beauty of this group is that it has 6 members, and all the members are highly diverse in function. And when I say it's highly diverse in function, it means that if you look at the first member of this group, specifically CCM1, it acts as a tumor promoter for pancreatic cancer. Whereas if you look at the CCM5, another member of the group, it acts as a tumor suppressor for breast cancer. So my work has been concentrated in this particular group of protein, CCM5, and so it's a small size protein, specifically 29 to 35 kDa size protein, and sorry for that. So my work has been concentrated on this CCM5 group of protein, and this is not new. It has been well documented. We have publications on this, that CCM5 works as a tumor suppressor in breast cancer models. But the thing that came into our mind while working in CCM5, that we know that if we increase the level of CCM5 or overexpress CCM5 in triple negative cells, you make it responsive to hormone therapies, because it gives you a property of ER alpha, or express ER alpha in the triple negative cells. But always doing or achieving the same with a recombinant protein or overexpressing it in a genetic level is not possible for a clinical setup. So we were looking for agents that would elevate the level of CCM5 in triple negative breast cancer cell. And then while looking at the literature and browsing through the literature, EGCG came across. EGCG is also not a new target, it's been there for so long. But the thing that captured our mind or captured our imagination is that basically it also has a tumor suppressor property to it, and it can also induce ER alpha expression in TNBC cells. So we thought that whether the expression of ER alpha in TNBC cells that is done through EGCG, whether it is somehow related to our gene of interest, that is CCN5. So what we did, first thing first, we did some expression studies. Basically, we treated cells with increasing concentrations of EGCG, and what we found that with increasing concentrations in TNBC cells as well as some ER positive cell lines in humans as well as mice cell lines, the level of CCN5 goes up. And we check the same thing at transcriptional level as well as translational level, and we check the same thing with whether it has the ability to bind to the receptor of CCN5, and we saw that it has the ability to bind to CCN5 and directly inducing the expression of CCN5. Then we did typical functional assays. We did some cell viability assays as well as so we did some cell viability assays as well as some apoptosis assays, and we found that EGCG has the property to induce apoptosis and reduce cell expression, increase expression of CCN5 in breast cancer or TNBC models. So we did some further assays with 3D assays, specifically sphere formation assays. We looked at the size of two separate cell lines, and after treating with EGCG, and we collected those pairs. We looked at the basic stimulus markers, and all the expression levels were going down. So till this point, we are very happy to see that okay, there is a molecule that can induce the expression of CCN5 in TNBC cells, and that particular molecule could be used for our purpose. But till that point, we were thinking about why not give it a next level and go for a targeted therapy with this particular EGCG. So we approached to our chemist collaborators, and we requested them whether we could coat these EGCG with Cartesian molecules and attach a PEG and a folic acid along with it. And they did exactly the same. So they basically made two molecules, one with the ECG nanoparticle inside it in the middle, and the folic acid and the PEG was on two separate sides, and the other molecule has a folic acid and PEG attached together with the nanoparticle. So we took these nanoparticles and did our functional experiments with it, and on their part they did some characterizations with the size of these nanoparticles. Along with those, took some pictures, and they had looked at the amount of drug that those nanoparticles carry. So while the first thing we have to look into is whether after all these modifications and nanoparticle coating, whether it's going through or it's doing its job as a target therapy. So we included these, put this drug into, got this nanoparticle into TNBC cells, incubated them for 24 hours, and we could see that the nanoparticle weight, particularly that has the folic acid and PEG attached to it on one side, had the highest amount of fluorescence to that particular cell type, a particular compound. So we went forward with this experiment. We did the same thing that we did with our normal compound. So we treated the cells with the same concentration of EGCG, and while doing so, we could see that normal EGCG is basically giving the same effect that has attached with the pegylated or folic acid attached nanoparticle coated EGCG that we have. Then we did some sphere formation assays as well as some colony formation assay, and on both cases we could find that it is giving us the same result that was being generated as just the chemical without any chemical modification or nanoparticle coating was able. So we took the entire experiment in vivo, and what we saw is basically it's repeating itself as what we see in vitro setup. And when we use EGCG for treating xenograft tumor models, subcutaneous xenograft tumor models induced with 231 cells, the tumor size went drastically compared to the control. But what surprised us is when we injected the nanoparticles in mice and tried to see whether it is giving us the same kind of result that EGCG is giving. But you could see the second bar is basically the nanoparticle, and it is not giving the desired effect that we have expected for. So that's a setback for this experiment, but in vivo it is in vivo, so it cannot go do whatever we want and what we expect all the time. So we kept it as an observation, and we are looking forward to work on this particular direction. So in conclusion, what I would say that this particular study has given us the direction that the EGCG can act as an agent that can promote CCN5 overexpression in TNBC cells, and this particularly nanoparticle coated or nanoparticle conjugated EGCG can act as a future target for used as a targeted therapy for TNBC cells or triple-negative breast cancer in human models. So this work had been published in Pharmacological Research Perspectives last year, and these are the mentors that I had from my postdocs, PhD supervisor to all the postdoc supervisors. These two are my just newly recruited two PhD scholars, and these are my collaborators that I'm currently trying to work with. That's pretty much it. Thank you very much, and I'll be happy to answer.
So any questions?
What are the side effects of EGCG? Side effect?
Side effects. So the main thing that when you treat with EGCG is basically at higher concentration this induces liver damage and as well as kidney damages in normal EGCG drug when you use it, because EGCG is responsible for inducing antioxidant effects, and too much of EGCG for prolonged time it induces kidney damage as well as septic damages.
Dr. Arnav, I have one question.
Yes, sir.
If you develop preclinical tumor model and then use combination of recombinant CCN5 along with EGCG, what will happen?
So theoretically, what the last slide I have shown is that when you use just recombinant protein, it has a tumor suppressor property too. Now, if it is in combination with EGCG,
it will give a better effect than CCN5 alone or EGCG alone.
For the last slide, obviously it will give us a better effect in combination therapy. But the main problem with recombinant CCN5 is obviously to procure it, to make a recombinant protein must have. That's the main hurdle, because we have done a lot of experiments with recombinant protein in this particular field, and it's a mess. So I don't want to go to that route in future to treat cells with recombinant proteins, and that's not a viable option though. But that's why we are looking for agents that can induce the level of CCN5 in tumor cells, basically.
I know there are some clinical trials with EGCG, right?
Yeah.
And do you know what does it do on CCN5 patients? Your EGCG?
CCL5 patients means
There are some clinical trials already were done on with EGCG. So my question is that is there any report what does EGCG does to the CCN5 in patients?
no, sir. There is no report so far that looked at that aspect. But yes, there are many trials going on EGCG.
Dr. Anand, this is regarding CCN5. You said that it is a tumor suppressor gene, right? It is like downregulated in DNBCs. So what's the mechanism? Is it methylated or it is transcriptally suppressed?
It's a ER responsive gene basically.
Okay, so here is not there, that's why.
No. So
Well, thank you, Dr.
thank you.
So now we move on to